Okay. What is the lymphatic system? Well, the lymphatic system, if we look at our diagram over here, to the spotlight, is in green. And it is closely associated with the cardiovascular system. The cardiovascular system, if you remember the arteries and capillaries and veins, it's no coincidence that the lymphatic system begins with the lymphatic capillaries, and it continues in a one-way circulation all the way back to the subclavian vein in the cardiovascular system. So again, this is closely associated with the cardiovascular system. The functions of the lymphatic system is to transport excess fluid away from interstitial spaces. Interstitial spaces means fluid that is not in a vessel, so fluid that is around tissues or cells. It's also called tissue fluid. It transports this fluid back to the bloodstream. And also, when we talk about digestion, we'll learn how the lymphatic system transports fats, and it helps defend the body against disease. This is a very nice representation of the lymphatic system, again in green. You'll notice these little nodules here. What these are, there's one right here. These are lymph nodes. And again, these act as filters of the lymphatic system. A few other parts of the lymphatic system that I'm not going to discuss today, but just you need to know, be cognizant of, is the red bone marrow, which is in the skeletal system. But the red bone marrow is where red blood cells and white blood cells. And again, one of the functions of the lymphatic system is immunity. So white blood cells are essential to that. Another is the spleen. This lymphatic organ would be in this general vicinity. The spleen contains white blood cells, and it also contains a lot of red blood cells, so it does a lot of filtering, and sometimes red blood cells go there to die. And we have the thymus gland, which is, well, this is the heart, of course, but the thymus gland would be up in this area, and we're going to learn that that's where the lymphocyte, one of the lymphocytes, the T cells, mature there. Okay, let me remember to erase drawings. So what is lymph? Lymph is the fluid of the lymphatic system. Again, the lymphatic system, the lymphatic capillaries are in green, whereas the cardiovascular capillaries are in purple. And if you remember the concept of bulk flow, for oxygen and other nutrients to get to the tissue cells, and let me click on the pointer here. No, pointer, here we go. So again, here are the cardiovascular capillaries. Here are the lymphatic capillaries. And bulk flow was how the capillaries, or how the nutrients of oxygen, glucose, amino acids leaked into, or got to the tissues, cells. And then the tissues in return carbon dioxide and other waste to the blood vessels. Also with bulk flow. Again, bulk flow is driven by osmotic pressure or hydrostatic pressure gradients. Well, bulk flow works or pumps out more fluids on the arterial side than the venous side. So that means the tissue fluid starts to increase not only in volume but also in pressure. And that pressure causes the tissue fluid, or the interstitial fluid, to go into the capillaries, or the lymphatic capillaries. Notice the arrows all go in one way. Once the fluid enters the lymphatic capillaries, it is now lymph, and it will travel all the way through the lymphatic system and eventually end up back in the cardiovascular system. So there's not much difference between lymph, blood plasma, interstitial, or tissue fluid. The only difference is location. And again, one of the reasons why the lymphatic system is so important is because you're removing excess fluid from the tissues. If you did not, you would have swelling or edema. Now, as for the flow of the lymphatic system and the organs of the lymphatic system, I think you guys are quite capable of learning that on your own. What I'm going to cover is the body's defenses against infection. So I'm going to cover the immunological portion of this chapter in some detail. First, we're going to learn what a pathogen is. Also, I want to mention before I get too far, and if somebody does join me, please feel free to type in a question, and I will answer it when appropriate, or when I can. Questions are always appreciated and appropriate. Pathogens, let's talk about what a pathogen is. A pathogen is a disease-causing agent. These agents can be bacteria, viruses, complex microorganisms like tapeworms or pinworms, and spores of multicellular organisms. These are things like fungus, and that causes athlete's foot, and ringworm. Now, not all of these bacteria, viruses, microorganisms, or spores are disease-causing agents, and that's a good thing because we would be dead if they were. What resistance and immunity is referring to is the ability to ward off the diseases through our defense system. So what we're going to be talking about is how our body defends against pathogens. We have two systems to destroy pathogens. One of them is innate, and these are just general defenses. They try to keep out and destroy as many pathogens as possible. They don't particularly recognize anything other than foreign matter. Adapted defenses, this is known as immunity, and this is more specific. So you might hear it is called adaptive or specific immunity. And it is targeting precise and specific antigens. An antigen is a protein on the plasma membrane. And again, this is very precise. If you might remember a few years ago when you went to get the flu vaccine, you could get seasonal flu vaccine or you could get the H1N1 vaccine. The H1N1 vaccine would recognize only the H1N1 flu. It would not recognize the seasonal flu. And the seasonal flu vaccine, in turn, would only recognize seasonal flu antigens, not the H1N1. Again, lymphocytes, you might have heard of T cells and B cells. These are the cells that are going to carry out immunity. In other words, these are the cells that are going to bind to the antigens. The first line of defense, so again, we're going to talk about innate immunity and go into a wee bit more detail. We're going to talk about physical barriers first. We have the epidermis. These are closely packed keratinized cells. Periodic shedding, as you know, if you have to dust your house every now and then. Remember, the epidermis is made of stratified squamous epithelial tissue. Again, the top layer you lose, and the bottom layer gets replaced by the bottom layer. So you're constantly replacing your epidermis. You have mucous membranes. Mucous membranes refer to membranes that open to the outside. These are membranes found in your nasal cavity, your oral cavity, vaginal cavity, anal cavity. And mucus quite common this time of year, helps to trap microbes and other foreign substances. Nose hairs trap and filter dirt and debris particles. Yes, they can be unsightly, but yes, they are functional. And cilia is found in the upper respiratory tract. For you histologists out there, if you remember pseudostratified columnar epithelium, some of those were ciliated. And again, the cilia moves particles along its cell surface out the respiratory tract. Oh, I keep forgetting to erase. Okay. Fluids that the body produces. You have the lacrimal apparatus of the eye. That's referring to the tears. Tears wash away pathogens. Tears also include lysozymes. These are special enzymes that break down bacteria cell walls. They're present in other fluids like saliva and perspiration. Saliva washes out the mouth. Urine cleans the urinary system. And vaginal secretions clean out the reproductive system. And defecation and vomiting, these clear out the digestive system. Chemicals that our body makes, our sebaceous glands secrete sebum, and that has protective properties, as does perspiration, gastric juices, and vaginal secretions. All of these are acidic, so they can kill bacteria because altering the pH can damage bacteria and other pathogens. Other things that are internal defenses, you have interferons. These primarily prevent viruses from spreading, and they do so by stimulating phagocytosis. Defensins, they cripple microbes. They're going to stimulate neutrophils. We're going to talk about neutrophils. These are phagocytes, cell eaters. Collectins are proteins that protect against a broad range of things, bacteria, yeast, and viruses. Complement proteins, these are a cool set of proteins. These stimulate inflammation, attract phagocytosis, enhance phagocytosis, and they can also poke holes in pathogens. It's kind of cool, and we'll show you this in a few slides. Iron binding proteins. Iron is important for growth of bacteria, so without iron, bacteria don't grow. And you also make antimicrobial proteins. And again, that gets rid of bacteria. Also, antimicrobial proteins can attract dendritic cells. We're going to learn more about these when we talk about T cells and B cells, or the lymphocytes, specific immunity. Mast cells secrete things such as histamines, which, as you know, if you're taking any allergy medicines, it's an antihistamine, and this causes an increase in inflammation, another type of immune response. Natural killer cells. These are lymphocytes, so these are a type of white blood cell, but they're not T cells or B cells. In other words, they're not specific. They kill a wide variety of infected body cells. They can make holes because they use a protein called peripherin, and peripherin makes perforations or holes in plasma membranes of pathogens, and they have granules or granuzymes. Granuzymes induce apoptosis, and if you remember, apoptosis is programmed cell death. Another defense mechanism are phagocytes. If you remember phagocytosis, that's cell eating, these two white blood cells, neutrophils, and monocytes, which will become macrophages when activated, are cell eaters. And there's 5 easy steps in phagocytosis, and I'll show you that in this slide. And I'm just going to click through, and then I'm going to go over it. The first step is chemotaxis, and that's referring to these little particles here. This is your microbe. This is the foreign guy that you want to, or the pathogen, I'm sorry. This is the pathogen that you want to get rid of. Here is the phagocyte. This chemotaxis are chemicals that are attracting the phagocyte to the microbe. Now here, the second step is called adherence. Adherence refers to the pseudopods, which will engulf, the pseudopods are kind of extensions of the phagocyte, and they're going to engulf the microbe. Once the microbe is taken into the cell via vesicle, that's called ingestion, then it meets up with a lysosome. Lysosomes are organelles that contain digestive enzymes. So you start digesting the microbe in step 4, and that leads to the killing of the microbe, and your phagocyte disposes of said microbe. Inflammation. This is another important nonspecific defense mechanism. Again, nonspecific means innate. There are 4 signs you may be familiar with. Well, actually, let me highlight those. Redness, pain, heat, and swelling. The redness, pain, heat, and swelling come from this stage here. Vasodilation. Vasodilation is referring to the enlargement of the lumen of the arteries. So you're getting more blood flow, and that's going to lead to redness, and also it's going to lead to heat. In addition, the capillaries in the damaged area become more permeable, so that means more fluids can leak out. That's going to cause the swelling and possibly the pain if there's pressure on the nerve. But the permeability, the leaking of the fluid, is important because remember that fluid has not only immunoglobulins, which are released by B cells, but it also has neutrophils and macrophages. Once these guys leave the capillaries, which is called immigration, they go to the damaged area and they begin to get rid of the foreign matter. Tissue repair, because you have more blood flow, now you also have more nutrients and you can begin to repair the damaged tissue. Fever. This is basically an elevation in body temperature. This does 2 things. It inhibits microbial growth, and also it increases the activity of phagocytosis to a point. Fever can be caused by a substance called interleukin-1. Interleukin-1 is a cytokine. We're going to learn more about these cytokines. Their job is to stimulate other parts of the immune cells. Particularly, they stimulate lymphocytes. And lymphocytes, again, are T and B cells. And one of the things that's interesting about interleukin-1 is that in addition to stimulating lymphocytes, it can also produce fever, and that fever in turn can inhibit microbial growth and increase phagocytotic activity. Let me erase here. This is a nice chart showing you the first lines of defenses. The first line would be your mechanical barriers, your skin and mucous membranes. You're trying to prevent the pathogen from entering the body. If the pathogen should happen to enter the body, we're now down here, what happens are your second line of defense, and we talked about these: the chemical barriers, natural killer cells, inflammation, phagocytosis, and fever. Now we're going to come over here and talk about the third line of defense. The third line of defense here, this is your specific or adaptive immunity. And you have two types: the cellular immunity, and for this we're talking about T cells. And you have the humoral immune response. Humor is kind of an older word for fluid. So these are B cell response, which are antibodies. Antibodies travel through the blood plasma, hence they're humoral. Okay, so again, adaptive or specific defenses are the third line of defense. In addition to being able to respond to a particular pathogen, this type of immunity is to determine self from non-self. In other words, self is you, and non-self is a potential pathogen. When this goes awry, you get disorders, autoimmune disorders, like myasthenia gravis or lupus, disorders where your own immune system attacks you. In addition, specific immunity produces memory cells. Memory cells are for later. You have active cells that fight the pathogen right now, but the memory cells are for if you encounter that pathogen later. Again, antigens are the molecules that elicit immune response. Antigens are the proteins found on the surface of the pathogens. Well, they're not necessarily proteins. They can be polysaccharides, glycoproteins, or glycolipids. But the point here, what I'm showing you in red, antigens are on the surface of the pathogen. And these provoke an immune response. So here we see an antibody binding to one of the antigens. The most effective antigens are large and complex, meaning they're fairly big proteins. So we're going to learn that sometimes to activate the immune system, we have special cells that chop up these antigens so they're more easily recognized by the T and B cells. Okay. Let's talk a little bit more about T and B cells. They originate in the bone marrow, both T and B cells. Again, these are lymphocytes. Remember, in the bone marrow you have pluripotent stem cells that can give rise to all blood types. This, including the lymphocytes, include the neutrophils, but also red blood cells and the cells that become platelets. B cells complete their development in the red bone marrow. So I like to B for bone marrow. T cells complete their development in the thymus. So thymus, T for T cell. There's two types of T cells. The first one is the helper cell, and the helper cell is also known as the CD4. CD4 is a protein that the T helper cell displays. Cytotoxic T cells are known as CD8. Again, this is a protein that the cytotoxic T cells display on their cell membranes. And cytotoxic, that's kind of a cool word here. Cyto meaning cell, toxic meaning, of course, killer. So these are the cells, cells that are actually going to destroy the pathogen, if you will. The T helper cells are going to stimulate not only the cytotoxic T cells, but the B cells. So they're going to help improve the overall immune function. Immunocompetence, that's referring to the ability to carry out an immune response. And in order to carry out an immune response, your T and B cells have to have antigen receptors. And let me try this. So the receptors are found on the T and B cells. Yes, I know that's kind of blocky, but the pen doesn't work real well either. And again, but these receptors are needed to identify the antigen. And if you remember, the antigen is on the pathogen. Oh yes, this is not going to work either. All right, I'll just abbreviate it, PATH. Okay. So again, let's try it this way. Spotlight. Again, the antigen receptors, these are on the T and B cells, and the receptors are necessary to identify the specific antigen. And the antigen is on the pathogen, the bacteria, the virus, whatever pathogen. Oh yes, let's definitely erase that. So when we're talking antigen receptors, we're talking proteins. Okay, where did my little arrow go? Here it is. Antigen receptors are proteins. So because of that, they can undergo genetic recombination. In other words, you can mix the A, G, C's, and T's and make a number of different types of proteins, and billions, in fact. Or now, two other proteins that are needed on the lymphocytes are these, the major histocompatibility complexes, or MHC, which is how I like to refer to them. They are necessary for the T cells to recognize foreign from self. You have two classes of MHC proteins. Class 1 is on everything but the red blood cells. So all body cells, all your body cells contain class 1 MHC. And this is also what helps when you're doing transplants. Not everybody's MHC1 are the same. So this is where you could get transplant rejection. You need to kind of match up the major histocompatibility complexes. Class 2 is only found on antigen-presenting cells. Antigens can be very large proteins. So in order for the T or B cells to recognize them, they need to be kind of chopped up. And that's where the antigen processing cell comes in. It ingests the antigen and it places it next to either an MHC1 or MHC2 molecule on the plasma membrane. Some antigen-presenting cells include dendritic cells. Again, these are found in the epidermis and in mucous membranes. Macrophages. These are phagocytes. And then B cells. Some B cells can act as antigen presenter cells. Let me click through this. Okay. Here we have an exogenous antigen. Exogenous means it comes from outside your body. This could be like the cold virus. So, let me put on my magic spotlight. So you engulf the antigen, and then you digest it into smaller fragments. Those smaller fragments are mixed with the MHC2, which are in the green molecules. They're pushed together, and then they're presented on the plasma membrane. Here, the antigen receptors on T or B cells can recognize the antigen now. Here it was too big, and now it's just right. Another type of antigen is an endogenous antigen. Endogenous means it is from the body. So let's say the cold virus has started to manufacture things using your own DNA. Those would be endogenous antigens. So they're large. So the antigen presenting cell takes it in, breaks it into small parts, melds it with MHC1, puts it together, and then packages it on top of the plasma membrane. So you've taken this large antigen and you've broken it down to a smaller fragment. And this fragment is one that either the T cell or B cell receptors will be able to bind to. And again, this is important because unless the T cells or B cells bind to an antigen, they're not activated. So let's talk about T cells and the immune response. And just to repeat myself, you have to activate the T cells. T cell activation requires the antigen presenting cell. Okay. Now, we'll come back to this bullet point in a minute. There are two types of T cells. You have the T helper cells. Again, these produce cytokines and other chemicals that are going to stimulate the cytotoxic T cells. These are the killer T cells, and also the B cells. So cytokines are a group of proteins, and I believe I have a little chart in my next slide, that can do a whole bunch of different things. But the big thing they do is stimulate lymphocytes. We've already talked about one cytokine. That was interleukin 2, or interleukin 1, that can cause fever. But interleukin 2 is the one we're going to be talking about the most. These are going to control lymphocyte differentiation and growth. And you can think about interleukin 2 as giving your car some gas. For your car to get anywhere, you need a little gas. And that's what the interleukins help do. So here we're going to talk about the CD4 cells. The CD4 cells become T helper cells. So let me highlight that. T helper cells are also referred to as CD4. They have to recognize an antigen fragment. Okay. After they recognize, so in other words, the receptors on the T helper cells have to bind to the antigen fragment with the MHC2 complex. Once that is bound, then they undergo clonal selection. So if we're going back to the car analogy, the T helper cell binding the antigen fragment is like putting your key in the ignition. Clonal selection is going somewhere. A clone is a copy of yourself. So this one activated T cell goes and proliferates. And one of the places it proliferates is the lymph nodes and other lymphatic tissue. This is why your lymph nodes may swell when you have an infection. They're swelling because you're increasing the number of cells. Now, once you are proliferating, your CD4 cell or T helper cell proliferating, you can make active T helper cells, and active T helper cells will secrete interleukin 2. And virtually all immune responses need interleukin 2. So the cytotoxic T cells and the B cells need interleukin 2 in order to operate fully. Another type of cell activated helper cells make are memory cells. These are not active at the time, but should the antigen appear again, these guys can quickly proliferate and differentiate. Okay, here is your inactive T helper cell. And we're going to go to this drawing down here. On the inactive helper cell, you have TCR, which is the receptor for a particular antigen, and you have the CD4 protein. Remember, the CD4 is the protein that determines that this is a helper cell, and all helper cells display it. Up here is the antigen presenting cell. Notice it has a fragment, and that fragment is presented with an MHC2 complex. Once this occurs, once you put the lock in the key, you get to activate the helper cells, and they'll go and either make active T helper cells, again, which secrete interleukin and other cytokines, or you make memory cells should you come into contact with that pathogen later. CD8 cells, the cytotoxic T cells. Oh wait, I wanted to highlight, not spotlight. So the cytotoxic T cells are CD8 cells. They recognize antigen fragments combined with MHC1. And so in addition to binding to an antigen, here we have the presence of interleukin-2. So for the cytotoxic T cells to become activated, they need interleukin-2. So you can kind of think of this as the gas to get going. Then they undergo clonal selection in a lymphocyte or another lymphocyte in a lymph node or another lymphatic organ. And then the active cytotoxic T cells, they attack not so much body cells, but foreign cells. And then you have the memory cytotoxic T cells. These are for later. For goodness sake, I keep forgetting to erase my drawings. But I've lost my cursor. Here we go. So here, let me get my highlighter out, or spotlight. Here is your inactive cytotoxic T cell. We're going to follow the yellow arrow. On that T cell, in orange, is the receptor for an antigen and the CD8 protein. This signifies that this is a cytotoxic T cell. Okay, and if we go back up here, we have the infected body cell. And actually, this infected body cell is an antigen presenting cell, and it has presented an antigen, and we're going to move back down here. And this antigen fragment is connected with the MHC1 protein. Okay, so now we have the lock and key. So your key's in the ignition. To get this inactive cytotoxic T cell to go, you need to give it gas. And that is the interleukin-2 stimulated or produced by the T helper cells. So once you have the interleukins, the gas, but first you got to have the lock, or the key in the lock, or the key in the ignition, the gas, then you have an activated cytotoxic T cell, and that helps form, after clonal selection, you get a whole bunch of activated cytotoxic T cells, and you get some memory T cells, and this is for later. Now, this is a little bit of an aside, but this helps me remember which CD protein binds with which MHC protein. I like to call it the immunological octet rule. So if you take 8 and you multiply it by 1, you get 8. So the CD8 protein needs the MHC1. And over here, you have CD4, and you have the MHC2 a protein. So 2 times 4 is 8. So the T helper cells need the MHC2 complex to present the antigen fragment. Nope, wrong button. And let me erase that arrow. Okay. Oh, actually, erase all drawings. Now I get to put the arrow on. How do cytotoxic T cells kill other cells? They kill like natural killer cells. But the main difference, and let me highlight that, cytotoxic T cells have a receptor. Excuse me. Whereas natural killer cells destroy a wide variety of cells. Natural killer cells are innate or nonspecific. T cells are specific or adaptive immunity. However, they're both lymphocytes and they both kill the same way. They can induce apoptosis or they can cause cytolysis by poking holes in the membrane of the pathogen. Now we have the B cell immunity. And activation and clonal selection of the B cell is quite similar to the T cell. First, an antigen has to bind to the receptor on the B cell. Now B cells are a little different. It could be unprocessed. And the difference between an unprocessed and processed antigen is size. Unprocessed antigens can be quite huge. Although B cells can also bind to a processed antigen or an antigen with MHC2 or antigen fragment combined with an MHC2 fragment. Also, interleukin-2 or other cytokines can also co-stimulate the B cell. And when you get interleukin-2, you get a much more intense process or response to the antigen. The B cell undergoes clonal selection, and again, clonal selection is proliferation. And proliferation means it makes more. We have a new term here, plasma cell. Plasma cell is the active B cell, and the plasma cells are going to secrete the antibodies. Memory cells, these guys do not secrete antibodies yet. They would should the antigen reappear. What this illustration is showing you is here's your B cell. It's active because it is bound to an antigen, and it can be a non-processed antigen. Also, notice here's some cytokines from T helper cells, and now it's proliferating. And when it proliferates, it can become a plasma cell, which will secrete the antibodies. Also, it will become a memory cell, which will be dormant for later. And this side is just showing you the same thing. This is the clone. Even though this particular illustration can be quite confusing, notice you have your B cells, you have your helper T cells, and your cytotoxic T cells. To get the optimal immune response, all three of these lymphocytes, all three lymphocytes have to be activated against the same pathogen to get optimal immune response. And down here in the picture, you can see T helper cells and phagocytes. So our immune system all works together. Let's talk about antibodies. Again, they're secreted by the B cells. And B for B cells, to be an antibody. These are immunoglobulins. These are proteins or gamma globulins. These hang out in the blood plasma. They have a variable region and a constant region. The variable region is what's going to bind to the antigens. It's variable, so it can bind to a wide variety of antigens. The constant region, there are 5 classes, and each class kind of has a different reaction. These are the 5 classes. You have IgG. Ig is immunoglobulin. G is just the type. IgA, IgM, IgD, and IgE. These are listed from the most abundant antibody or immunoglobulin to the least abundant. And as you can see in this chart, gives your various functions. The IgG, again, the most abundant of all. This one can be passed through the placenta, so this is one of the ways a mother helps to pass immunity on to her unborn child. IgA, this is made by exocrine glands. One of the exocrine glands that makes this is the mammary gland. This is another way that a mother can help her child's immune system by passing antibodies to her nursing child. IgM, you've encountered this one when you've seen slides of blood typing. That agglutination or coagulation that occurs is due to the IgM antibody. IgD, this is an antibody. It's on the surface of most lymphocytes, and it can serve as a receptor. The IgE is also an exocrine gland secretion. So it promotes inflammation and allergic reactions. So I think this time of year in Indiana, IgE numbers are up. Or they at least they are for me. Okay, how do antibodies protect against pathogens? Well, one, let me get my handy dandy spotlight here. They can neutralize antigens. They can immobilize bacteria. Agglutination and precipitation. You saw that when we did blood typing. They ball up and they can't do anything. They can enhance phagocytosis. And then they can activate the complement system. And we've talked about the complement system before. In addition to causing phagocytosis and inflammation, it can also cause cytolysis. And that's triggered by this lovely cascade system. And what a cascade system means is that one protein stimulates another protein, and that stimulates another protein, and on and on and on. It's a cascade effect. Well, this happens to form something called the membrane attack complex. In other words, it's poking holes in the plasma membrane of a pathogen, and it causes the pathogen to lyse or break open. Immunological memory. Here we're dealing with the difference between the active lymphocytes and the memory lymphocytes. And this is kind of the basis for why we have vaccines. You have thousands of memory cells. So they're going to appear, so once the pathogen appears for a second time, they can differentiate between hours. So here's a primary response. So say you get a cold, and it takes a few days to get over a cold. The reason why, it takes a little bit of time for your antibodies to kind of be processed. Now once you're exposed to the cold virus, the next time you have a much larger concentration of antibodies, so a quicker reaction, so thusly you don't get as sick. Or if you get sick at all, your immune system is waiting for it. And again, when you get vaccinated, let's say with a chickenpox vaccine, your first exposure is to a type of virus like well, it used to be cowpox, but a dead virus, you get some immune response. But what you're trying to produce is the memory cells. So when you actually come in contact with a live chickenpox virus, boom, your immune system is ready to go. There's a few ways to get immunity. You can get, well, two types of immunity. You can get active immunity or you can get passive immunity. Active immunity revolves you actually making the antibodies and the active lymphocytes. So your body is actually doing this. And it can either be naturally acquired or artificially acquired. Naturally acquired, for example, we've been talking about chickenpox, is how I got the chickenpox. I got the chickenpox because my aunt happened to have the chickenpox. So my mom took me to my aunt's house so I could get chickenpox. Artificially acquired immunity is what's happening to my children. My children are now being vaccinated for the chickenpox. So now they're immune from the chickenpox because they have been vaccinated, not because they've been exposed to it. Passive immunity is when you're given somebody else's antibodies or activated lymphocytes. They can be artificially. For example, you're given an injection of gamma globulins. Say you have some sort of immune disorder where you're not making a specific type of antibody. Naturally acquired passive immunity would be from any immunoglobulins or antibodies that pass between the fetus and a pregnant woman, or when a woman is breastfeeding. That is naturally acquired. Okay. Oh, I believe that's it. So this is the end of my presentation. I'll be here for a few minutes to answer questions. I don't see that anybody's out there, but I have enjoyed myself, and I hope you guys have learned something. And anyway, I'm going to stop recording, and hopefully this recording will be uploaded on YouTube. So